Okay, hello to all of you. Let's discuss chapter number 10, part 2. So, in this part, we are going to discuss the details of light reactions and the Kelvin cycle. So, I'm going to. So, basically, in the light reactions, we have already talked about what are the main events which take place in the light reactions. So light reaction is called light reaction, or it's called a photopart. It's called light reaction or photopart because it involves sun's light. And some of the events which takes place in this part are: number one, the photolysis of water, the spreading of water and release of oxygen, and then there's conversion of ADP to ATP. That's what we're going to talk about. And the conversion of NADP plus to NADPH. So these are the main events which takes place in the light reaction. So in this, the energy is produced in the form of ATP and NADPH. And the second phase of the photosynthesis is Kelvin cycle, or synthesis phase, or dark reactions, or dark phase. In this phase, the ATP, the energy which is present in the form of ATP and NADPH, is converted into organic molecules. It's converted into organic molecules, basically. So that's what this Kelvin cycle is. So let's talk about the light reactions. So the light reactions involve two kinds of electron flow. One is linear, another is cyclic. Okay, and we are going to talk about what is the outcome of the linear electron flow and what's the outcome of cyclic electric flow. So we have already discussed about the photosystem. We talked about what photosystem is. It has a light harvesting complex and it also has a reaction center. And we have talked about the pigments which are present in the light harvesting complex and the special pair of chlorophyll molecules which is present in the reaction center, which is P680. And the energy is transferred from the energy which is coming from sun, is transferred through the pigments. It reaches the two special molecules of chlorophyll A, and that energy helps in the exciting of electrons in the chlorophyll A molecules, and those electrons then they are taken by the primary electron acceptor in the reaction center. And then let's see what happens to the electrons after that. So the output of the linear electron flow is ATP and NADPH, and it involves two photosystems. Okay, we just discussed in part 1 that there are two photosystems, photosystem 2 and photosystem 1. Photosystem 2 is also called P680 because this photosystem absorbs the pigments in this photosystem, the special pair of chlorophyll A molecules. It absorbs the light of wavelength of 680 nanometers. And the photosystem 1, it absorbs the light of the wavelength of 600 nanometers. So there are 8 steps in the linear electron flow. So let's talk from here actually. Okay. So this is basically the linear electric flow. Okay. So there are two photosystems involved. The photosystem 2, photosystem 1. I just explained what photosystems are. Okay. And in between these two photosystems, there is a electron transport chain. So electron transport chain we have already covered in the chapter number 9 about cellular respiration. What is electron transport chain? It is basically some of the protein complexes embedded in the cellular respiration. We talked about protein complexes embedded in the inner membrane of mitochondria. But here in photosynthesis, the electron transport chain is some set of protein complexes which are embedded in the thylakoid membrane. Okay, so this is the electron transport chain, which is some protein complexes which is present. These protein complexes, they are embedded in the membrane, thylakoid membrane, between these two photosystems. And after this photosystem 1, there is another protein here which is called ferridoxin, and it makes it makes a very short electron transport chain which is after this photosystem 1. Okay, so basically this is photosystem 2 This is the light harvesting complex. This is the protein with these pigments embedded on it. This is the reaction center, the special chlorophyll A molecule, P680, and this is the primary electron acceptor. This is photosystem 1. This is the light harvesting complex. This is the protein with these pigments, and this is the reaction center with these special chlorophyll A molecule with the which can absorb the wavelength of 700 nanometers, and this is the electron acceptor, primary electron acceptor. So basically these pigments which are there in the light harvesting complex, they absorb the light from, they absorb the energy from sun, okay, and this energy is passed through or transferred through these pigments, and this energy eventually reaches this special molecule of chlorophyll A, and the electrons, and that energy basically excites the electrons in this pigment, and then these electrons are accepted by this primary electron acceptor. And as a result of that, what happens when these electrons are accepted by this primary electron acceptor, this P680, this chlorophyll A molecule, it becomes oxidized. So if this becomes oxidized, okay, and electrons reaches here, and eventually you will see that these electrons are passed to this electron transport chain, which is between these two photosystems, okay. So when the electrons are passed here, it means there will be shortage of electrons here, because this chlorophyll has already lost the electrons. It has already lost the electrons to this primary electron acceptor, and this primary electron acceptor passes electrons to this electron transport chain. So to prevent this loss of electrons, another event takes place, which is the splitting of water, or it's called photolysis of water. Why it's called photolysis? Because the splitting of water takes place in the presence of sunlight. So this water molecule, it breaks, it splits, and it produces oxygen, and this is the oxygen which is the product of photosynthesis. This is the oxygen, this is the molecular oxygen which the plants are, or the photosynthetic organisms are giving out. And then in water, there are also hydrogen atoms. So those hydrogen atoms, they give protons as well as they give electrons, okay, and these electrons are given to this chlorophyll A molecule, okay. So these electrons which are coming from hydrogen atoms, they're given to this chlorophyll A molecule, and there is a continuous supply of electrons, okay. Because this chlorophyll A molecule loses electrons to this, it loses electrons to this electron transport chain. To have the continuous supply of the electrons, water splits, and the electrons come from hydrogen atom, and these electrons, they are given to this chlorophyll A molecule, and as such, and because of that, there is a continuous supply of electrons, okay. So when so there's a continuous supply of electrons to this electron transport chain. So this electron transport chain is between these two photosystems. So now let's see what is happening here in the photosystem 1. The same thing is happening in photosystem 1. It has the light harvesting complex, it has this reaction center. This light harvesting complex has pigments embedded on it. So they basically take the sun's light, okay, sun's energy, and this energy is passed through these pigments, and it eventually reaches this special chlorophyll A molecules, pair of chlorophyll A molecules, and this is called P700 because they can absorb the radiation, the light of 700 nanometers. So same thing is happening, these electrons passing energy, the electrons getting excited here, and then they are given to this primary electron acceptor, and from this primary electron acceptor, these electrons are passed to a protein called ferredoxin, which is basically just one protein complex here, and which makes a very short electron transport chain, okay. So same thing is happening here, that this chlorophyll A molecules is losing electrons to this electron acceptor, and this electron acceptor is passing electrons to this protein, to the ferredoxin. Again, this chlorophyll A molecules, it gets oxidized, okay. So to have the continuous supply of the electrons, because this loses electrons to this. Okay, so same thing is happening, what was happening here. So so from where does this chlorophyll molecule is getting the continuous supply, as this was getting from the splitting of water? This is getting the continuous supply from these electrons which are coming through this electron transport chain. Okay, so through this electron transport chain, electrons are coming, and it gives the continuous supply of electrons to this chlorophyll A. But remember that these electrons, I'm going to talk about these electrons, they're coming through this electron transport chain, and these electrons, they don't have a lot of energy in them. They are low energy electrons. So this is what is happening. So now let's talk about what is happening when these electrons from this photosystem, when they're traveling through these protein complexes. So when they're traveling through these protein complexes, so something very similar to what was taking place in the cellular respiration is taking place here. These electrons, when they pass through these protein complexes, these are high energy electrons. They are going to release their energy on the way, and that energy is going to create a proton gradient, just the way it was happening in cellular respiration. In the cellular respiration, proton gradient was being created. So the protons were being taken from the matrix region of the mitochondria, and these protons were being pumped to the intermembrane space. And as a result of that, proton gradient was being created, and these protons, they were flowing through a protein called ATP synthase, and as a result of ATP was being produced. So similar thing is happening here. These electrons, they're passing through these electrons, they're passing through this electron transport chain, and they're releasing energy, and that energy is being used to pump the protons from the stroma to the thylakoid space. Okay, and then as a result of that, proton gradient is created in the thylakoid space, and these protons, then they flow back through the protein called ATP synthase. They flow from thylakoid space. They flow from thylakoid space to the stroma, and ATP is produced. Okay, so this is the way ATP is produced by the process of chemiosmosis, just the way it was being produced in the cellular respiration. So this is a step how ATP is produced in the light reactions. And then now let's look here. These electrons which are being passed from the primary electron acceptor to the ferredoxin protein, these electrons, they are actually taken by the electron carrier called NADP+. So this is the electron carrier which is involved in the process of photosynthesis. So it takes the electrons which are coming from here, and it gets reduced to NADPH, and it takes place with the help of the enzyme called NADP+ reductase. So this is the way how in the light reactions ATP is produced and NADPH is produced. Okay, so these two energy forms, which is in the form of ATP and NADPH, they are further used in Calvin cycle, and in the Calvin cycle the sugar molecule is produced. So we are going to talk about that. So this is all what I just explained is written here, and now I'm going to, so this is a cartoon showing how ATP is produced and how NADPH is produced. So this is photosystem 2, this is photosystem 1. The electrons flowing through this electron transport chain produces ATP, and these electrons which are passed through the electron transport chain, which is at the end of the photosystem 1, those electrons are accepted by NADP+, and NADP+ gets converted into NADPH. Okay, so we just talked about the linear flow of electrons. We just talked about linear flow of electrons, and in the linear flow of electrons, two photosystems are involved, photosystem 1 and photosystem 2, and ATP and NADPH are produced. Now we are going to talk about cyclic electron flow. It's a kind of electron flow which takes place in some special kind of bacteria, and also it can take place in some phase of the life of the plants. When there is a lot of sunlight, then they convert from linear electron flow to cyclic electron flow. So in the cyclic electron flow, there is involvement of only one photosystem. In linear electron flow, there is involvement of two photosystems. In the cyclic electron flow, there is involvement of only one photosystem. And in the cyclic electron flow, there is only production of ATP and no NADPH is produced. and also no oxygen is released. So in the cyclic electron flow, basically the electrons after they are passed from the after they are passed to the primary acceptor, it involves only one photosystem 1. So the electrons, they are passed from primary electron acceptor to ferredoxin, which is a short electron transport chain after this primary acceptor. So from the ferredoxin, the electrons are not given to NADP plus, but instead these electrons, they are cycled back to the electron transport chain which is present between the two photosystems. So these electrons are given to these protein complexes, and then they through this protein they eventually come back and reach this photosystem 1. So when these electrons, they're flowing through this electron transport chain, there's a production of ATP. So in the cyclic electron flow, there is only production of ATP, no NADPH is produced, and there is only involvement of photosystem 1. And this protein is plastocyanin. So from ferredoxin, it reaches the electron transport chain which is present between two photosystems, and then through this protein plastocyanin, because this is the one, even in the linear flow, this protein gives the electrons to this photosystem 1. So the electrons keep on recycling here. So here is just the comparison of the process of chemiosmosis by which ATP is produced in the cellular respiration and the photosynthesis. So here you can see this is the matrix part of the mitochondria. This is the intermembrane space of the mitochondria. So as the electrons flow through the electron transport chain, the energy is released, and that energy is used to pump the protons from this region to this region. And then the electrons flow through ATP synthase and produces ATP. But in the photosynthesis, the proteins, they are pumped from stroma region to the thylakoid space, and as a result of that, there is an accumulation of more protons in this region and less in this region. And then these protons, there is a high concentration of protons here, this is less concentration of protons here, then they fall back from this region to this region through ATP synthase, and it results in the formation of ATP. So this basically overall shows you that how these photosystems are there, photosystem 2, photosystem 1, and in between these two photosystems, ATP is produced, and at the end of the photosystem 1, NADPH is produced. So ATP, so here you can see that this is the electron transport chain between the two photosystems. The electrons are going to flow through these proteins, and as a result of that, energy will be released, and that energy will be used to pump the protons from this region, stroma, to the thylakoid space, and as a result of that, there is a high concentration of protons in this region, and that results in the formation of a proton gradient, and then the protons, they flow or they diffuse from this region to this region. They flow from this region to this region through this protein, and that results in the formation of ATP. So ATP is produced in stroma, NADPH is also produced in stroma, and Calvin cycle also takes place in stroma, because these two forms of energy, they are produced in the stroma. And now the last thing we are going to discuss in this chapter is about the Calvin cycle, which is the anabolic process. It builds the sugar, and the energy is coming in the form of ATP and NADPH. And Calvin cycle involves 3 phases. First is carbon fixation, and it is catalyzed by a very, very important enzyme called Rubisco. And this is the most abundant protein which is present in our biosphere. And second step is reduction, and third step is regeneration of the carbon dioxide acceptor, which is RUBP. RUBP is ribulose bisphosphate. So I'm going to complete. So remember that this is Calvin cycle is a cycle. Okay. So remember that cycle means from where we start. at the same compound we end. Okay, so in the stroma there is an important compound present which is called ribulose bisphosphate. This is a compound with 5 carbons in it. 1, 2, 3, 4, 5. Okay, this is called ribulose bisphosphate. It has 2 phosphate groups and it has 2 phosphate groups and 5 carbons. So it's there. Okay, so carbon dioxide, which is there in the atmosphere, which is released in the atmosphere because of the process of cellular respiration, this carbon dioxide combines with this compound called RUBP, which has 5 carbons. So carbon dioxide has 1 carbon, RUBP has 5 carbons. So together they make one compound with 6 carbons. Okay, but this cycle operates 3 times to produce 1 molecule of sugar, and the sugar is glyceraldehyde 3-phosphate, which is intermediate metabolite in cellular respiration also. So this cycle has to operate 3 times to produce 1 molecule of glyceraldehyde 3-phosphate. So just to understand here, this RUBP combines with, so 3 molecules of RUBP combines with 3 molecules of carbon dioxide. Okay, and they produces, so how many, this is RUBP with 5 carbons, 3 molecules of carbon dioxide, in total 18 carbons. So, and this step takes place with the help of very, very important enzyme called ribisco. And the compound which is formed, has 6 carbons in it. Okay, 6 carbon compound is formed and 3 molecules of a 6 carbon compound is formed here, 18 carbons. Okay, it's a very unstable compound and it immediately breaks into a compound having 3 carbons and 6 molecules of that compound are formed. And the compound with 3 carbons is called 3-phosphoglycerate. Okay, 3-phosphoglycerate. Now this, in the next step, 3-phosphoglycerate is converted into a compound called 1,3-bisphosphoglycerate, which means it is getting the addition of 1 phosphate. From where does this phosphate is coming? This phosphate is coming from the ATP which was produced in the light reactions. Okay, so there were 3 molecules of this compound with 6 carbons. So they are unstable. They get converted into a compound with 3 carbons. So they are going to be 6 compounds with 3 carbons. 3-phosphoglycerate. This 3-phosphoglycerate, 6 molecules of 3-phosphoglycerate gets converted into 6 molecules of 1,3-bisphosphoglycerate. Addition of second phosphate. Because there are 6 molecules, 6 ATPs are going to be involved, and these 6 ATPs, they are going to be converted into 6 ADPs. After this, 1,3-bisphosphoglycerate gets reduced. Okay, remember in cellular respiration everything was getting oxidized. In this step is called reduction step, because this compound is getting reduced, which means it is accepting electrons. From where does these electrons are coming? These electrons are coming from NADPH. NADPH is produced in the light reactions. So 6 molecules of NADPH, they are going to give electrons to 6 molecules of 1,3-bisphosphoglycerate. And as a result of that, 6 molecules of glyceraldehyde 3-phosphate are going to be formed. Okay, so this compound gets reduced. Okay, 6 molecules of 1,3-bisphosphoglycerate, they get reduced and they form 6 molecules of glyceraldehyde 3-phosphate. Now out of these 6 molecules of glyceraldehyde 3-phosphate, plant is going to use only 1 molecule of glyceraldehyde 3-phosphate for itself. Okay, it is going to convert this glyceraldehyde 3-phosphate into glucose, sucrose, whatever the plant cells require. The other 5 molecules of glyceraldehyde 3-phosphate, the 5 molecules of glyceraldehyde 3-phosphate, they will be used to regenerate RUBP. RUBP is that 5 carbon compound which is there in the stroma all the time, and it is regenerated. It is regenerated from glyceraldehyde 3-phosphate. So out of 6 molecules of glyceraldehyde 3-phosphate, 1 is going to be used by plants, and 5 molecules of glyceraldehyde 3-phosphate, they are going to be used to regenerate RUBP. So glyceraldehyde 3-phosphate has 1 phosphate, then there will be addition of 1 more phosphate, and that is going to be with the help of ATP, and this glyceraldehyde 3-phosphate gets converted into ribulose bisphosphate. So ribulose bisphosphate is ready, and ribisco enzyme is ready for more carbon dioxide to come, combine, and this cycle operates, and this is the way organic molecule is formed. So this first step is called carbon fixation, because carbon is combining, this is called carbon fixation because here carbon dioxide is combining with RUBP. And then the second step phase is reduction, where this compound is getting reduced, and third is the regeneration of RUBP. So this is the Kelvin cycle. So this chapter finishes here. If you have any questions, please let me know.